It's a great honor and pleasure to lecture here and be accompanied by such an esteemed panel of moderators and speakers. Truly is an honor. So as we know, what we do in the laboratory is extremely important for maintaining embryo development. Several publications indicate that varying culture conditions can impact things such as blastocyst formation. And the further evidence for impact on embryo development by what we do in the laboratory can be seen in emerging literature, such as some of the abstracts listed here, where not only can embryo development to the blastocyst stage be impacted by various lab conditions, but also the act of mitosis, which makes sense, but as indicated by these publications, from examination of things like mosaicism. So the laboratory culture environment impacting mitotic aneuploidy, or varying rates of mosaic embryos between laboratories based on varying lab conditions. And so we know that some laboratories can grow blastocysts at higher rates than others. And in a similar fashion, these emerging studies on mosaicism in lab conditions indicate that sometimes mitotic errors can vary as much as 30% between facilities. And really what this means is that what we do in the laboratory absolutely can impact the embryo development. And as a result, we want to make sure that we are using optimized culture conditions in the laboratory to try to avoid doing damage and hopefully create a conducive environment to maximizing embryo development. And that really means trying to minimize the amount of stress that we're exposing our gametes and embryos to. I think most of us would agree that improper lab conditions can lead to environmental stress. As we've just mentioned, this can compromise cell function and development. And this is really important with gametes and embryos because they're especially sensitive cell types, especially those eggs and cleavage stage embryos. And so really this is why we study science. This is why we do the research that we do. And we're going to hear some of this research highlighted at this fantastic meeting. But considering the gamete and embryo physiology can help us combat that stress. This is why we're constantly all asking questions and doing research and presenting our findings in platforms such as this. So I'm going to talk a little bit about some common lab stressors: osmolality, temperature, mechanical stress, and pH, and talk about how this can impact the embryo in the lab to educate us a bit as to why we should be concerned about these stressors. But let's also importantly discuss steps that we can take in the laboratory to prevent these stresses. So, of course, I like to start my lecture with pH. A lot of emerging important data on pH with the impact on gametes and embryos. And as just a brief refresher, we know that the denuded mature oocyte lacks the ability to robustly regulate its internal pH. These transporters and porters aren't really activated until after fertilization. So the denuded egg is an especially sensitive cell type. Cryopreserved and thawed embryos are especially sensitive to changes in pH. They don't really gain or regain the ability to regulate their internal pH for about 3 hours after warming. And we know that sperm function and internal pH are also influenced by media pH. And so this really just goes to highlight why we are concerned about pH in the laboratory, because it can impact cell development and function. And so this is why in the laboratory we think of pH in three main phases. Equilibration, you want to make sure that your embryo culture media has been given enough time for the pH to equilibrate inside the incubator. And this does take time. Rule of thumb is at least 8 hours. Most people do this overnight. And then the set point of pH, or that ideal range of where we want the pH to be to optimize embryo development. And this is a point of debate, something that's still actively researched, and we'll talk a little bit about that. And then very importantly, stabilization of pH. And so this has become less of an issue with uninterrupted embryo culture and time lapse, but certainly as we handle our gametes and embryos and move them in and out of the incubator for various procedures, we want to make sure that we're taking approaches to help avoid oscillations or changes in that pH set point that can damage gametes and embryos. And so we talked a little bit about some prior studies that indicate the importance of internal pH on gametes and embryos. And this is just one example of other literature further highlighting the importance of media pH. This is an abstract from ESHRE from 2016, published by Marius Menkes and others, where they used a sibling zygote split, and they looked at varying pH paradigms. And they showed that not only did blast formation rates vary based on different pH, but also time-lapse morphokinetic timings. And again, this further goes to illustrate the impact of pH on the mitotic process, the time it takes for these cells to mitotically divide. And as a result, low to high pH paradigm with a pH change on day 3 yielded more blastocysts and faster morphokinetic timings than the control, which had no change in pH, and also from the high to low pH paradigm. Some more recent data from RBM Online further indicates the potential impact of pH on mitosis. This actually was done in the mouse oocyte, so this would be the meiotic spindle, but changes in pH changed the spindle retardants as indicated by pulse scope imaging of the meiotic spindle. And so it's feasible that a similar mode of action might be present on the mitotic spindle. And something like this may be potentially responsible for why we see changes in morphokinetic timings in other studies. So certainly pH can impact embryo development. It may be impacting mitotic divisions, and just goes to highlight why we should be concerned about this in the laboratory. There's various steps that we can take to stabilize pH. You can use an isolate chamber with the gassed environment. You can use heaps or mops buffered media to stabilize pH outside of the incubator. You can make sure you use sufficient oil overlay over your microdrops for things such as biopsy or ICSI, and then proper incubator management, meaning you have enough incubators and that you've spread your cases out enough that you're not constantly opening and closing the incubator door, which obviously can cause a change in CO2 concentration and the resulting change in media pH. So going hand in hand with the importance of pH, making sure that you have the right set point as well as stabilizing pH, really need to also then know your target pH. What range are you aiming for? And this is usually in the range of 7.2 to 7.4. To know that, you need to measure pH. And importantly, you need to measure pH accurately. Although we use the percent CO2 concentration as a proxy for pH, it's not always reliable. Not all media are made the same. Various additives to media can change the CO2 requirement. So measuring pH is recommended. And this can be done with a variety of methods. A very good method is use of a pH meter or a blood gas analyzer, which is useful for the very small volumes that we use, especially in modern benchtop incubators. There are also some emerging real-time pH sensors that are very nice if you can gain access to them. And then the key is to make sure that you monitor this regularly to make sure that your conditions are giving you the pH that you're looking for. So another stressor that we talked about in the beginning was osmolality, and certainly osmolality can absolutely impact embryo development. You can see in the graphs work from Jay Baltz's group, where if your osmolality increases too high, embryo development is inhibited. Now, we know that embryos and gametes can handle transient or controlled changes in osmolality, and this is observed most readily with the exposure to cryoprotectants and warming solutions, where you'll see an oocyte shrink up in response to a high osmolality vitrification solution. And if you then bring it out of that solution in a controlled fashion through your warming media, obviously we can have subsequent fertilization and embryo development. But it's when you have uncontrolled or unknown changes in osmolality, especially those that get above 300 milliosmolar, is where you can run into issues. This is why most media are usually in the range of 260 to 290, and most modern media are closer to that 260 range. And importantly, they include key amino acids. Amino acids help cells regulate their volume, which is, as we indicated, impacted by osmolality. So not only can osmolality impact embryo development, it can also potentially impact mitosis. And so here you can see an older study out of John Kreitzer's lab, work done with Steve Mullen, who's an excellent physiologist and cryobiologist. But the osmotic stress can impact the metaphase 2 spindle of human oocytes. For every 1 millimosmolar increase, there was an increased spindle disruption by an odds factor of 1.001 to 1.004. So what this means is that as osmolality goes up, we know that embryo development can be impacted. In this case, the meiotic spindle can be impacted. And so again, there could be a feasible connection to the mitotic spindle as well. And this could have a subsequent impact on mitotic divisions and perhaps explain why some laboratories see high rates of mosaicism or aneuploidy than others. And so just another reason to make sure that we pay attention to these stressors in the laboratory, because there could be very real impacts on the resulting embryos that we're producing. And so that being said, we want to make sure that we take precautions to avoid osmotic stress. And that really means taking steps to avoid evaporation. And this can occur primarily with two main procedures in the laboratory. That'd be dish preparation, specifically with microdrops, as well as the use of our incubators for extended culture, especially uninterrupted culture. So anything that can impact evaporation, like airflow, higher temperatures, increased amount of time, small volume, or lack of humidity can induce more evaporation. And as an example, this is a 4-well dish placed on a warm surface at around 37 degrees during a oocyte retrieval. There's no oil over the top of this dish because this particular laboratory, the cumulus cells around the oocytes after retrieval would sometimes stick to the media oil interface and they didn't like it. But after just 5 minutes, you can see condensation on the lid of the dish. And so that's an indicator clearly that evaporation has occurred, and the osmolality and the substrate concentrations in that culture media in that dish are not what they were originally out of the bottle. And so while they may not have reached detrimental levels, clearly this is a stressor. That media is not as it was produced by the manufacturer. And so evaporation can occur very quickly if we're not careful of those other variables. And this further highlighted from a publication that we published a few years ago out of Gary Smith's lab at the University of Michigan. And as we mentioned, preparing microdrops is an area where evaporation can occur. And so we looked at variables like the temperature of the surface that microdrops were prepared on, the presence or absence of airflow, how we made the microdrop, as well as the volume of the microdrop. And 3 of those factors had significant impacts on resulting evaporation and osmolality increase. And all 4 of those variables had a significant interaction. And as a result, there was a greater than 40 millimolar osmolar increase in the osmolality of the media based on the best preparation conditions and the worst preparation conditions. And so depending on your starting osmolality, 40 milliosmolar could easily put you above that 300 milliosmolar point where we know the embryo development starts to become compromised. Uninterrupted culture, as you mentioned, is another area. Certainly this is being more widely adopted, especially with the use of time-lapse. And more incubators are dry incubators without humidity. And there's a few benefits to that in terms of contamination and other variables. But as a result, evaporation can occur if you don't take precautions. And so this is not to say that uninterrupted culture cannot work well. And it's not to say that dry incubators cannot work well. They absolutely can. But if you don't take the proper precautions, as you can see here in a dry incubator indicated by the light blue bars, significantly more evaporation occurred under these particular conditions of 25 microliter microdrops under 3 milliliters of oil. And as time went on, more evaporation occurred, perhaps not surprisingly. And by the end of 6 days of culture, 7 days, if you include equilibration, you can see that we're right near or above that 300 milliosmolar benchmark. So again, in this particular case, adjusting oil volume or oil type, or looking at other variables could help reduce this amount of evaporation and avoid compromised embryo development. So some practical tips in terms of osmolality and evaporation: utilize media with key amino acids. Most media now have these. Prepare your dishes carefully, as we mentioned. Importantly, limit the number of dishes you make at a time to avoid evaporation occurring. Keep media and dishes capped or covered as long as possible, especially if oil is not being used. Ensure you have proper humidity if using no oil overlay. Try to use humidification if possible. Consider the amount of oil you use as well as the type of oil. Heavier oil can reduce the amount of evaporation, especially in dry incubators or uninterrupted culture. Measure osmolality. If you can get an osmometer to help validate your system and make sure that your osmolality is what you think it is and that evaporation has occurred, and also making sure that you understand what the starting osmolality of your is of your media. If your media starts out at 280 or 290 milliosmolar, you have much less wiggle room for evaporation to occur than if your media starts out at 260 or 270 milliosmolar. So related to both pH and osmolality is temperature. And we all know that we do our best to maintain temperature in the laboratory for various reasons. Temperature can impact pH, especially in buffered media. Temperature impacts spindle dynamics, and certainly we would know that the spindle would control separation and segregation of chromosomes and can impact potential mosaicism or aneuploidy. Interestingly, it estimated that the temperature in vivo in the follicle is a few degrees cooler than core body temperature. And there's other animal data suggesting even a potentially cooler temperature in the female reproductive tract than core body temperature. And so this could have some implications on the optimal temperature that we would use in our laboratory. We all aim for around 37 degrees, but this in vivo data suggests that perhaps something closer to 35.5 or even 36 degrees may be more physiologic and perhaps better for gametes and embryos. It's important to note that when you talk about temperature, there could be some differences between the impact of an extended change in temperature. If you're going to culture something at 36 degrees for 5, 6, or 7 days versus periodic deviations. And certainly the periodic deviations are what we would experience most in our lab, moving things between microscope stages and hoods and incubators. And I think both are important, but there may be a different impact based on those paradigms. And so we had done some work in our laboratory with a novel time-lapse incubator with 6 individual chambers where we changed the temperature in each chamber by ½ degree. And we showed that absolutely embryo development was impacted in the mouse model. As the temperature went up or down from 37 degrees, we saw impacts on blastocyst development. And interestingly, we also saw impacts on morphokinetics. And perhaps not surprisingly, as the temperature increased, mitotic cell divisions got faster. And not necessarily as a better indicator because blastocyst development went down, but certainly temperature can impact mitosis, morphokinetic timings, and embryo development. And so we do want to be aware of the temperature we're using and avoid deviations in temperature if possible. And there are now at least 2 peer-reviewed publications looking at the impact of different temperatures on extended embryo culture in the human. And this is from Richard Scott's group out of New Jersey in the United States, where they looked at comparing 37 degrees Celsius to 36 degrees Celsius. And they certainly showed that 37 degrees Celsius gave better development on day 3, higher blast development rates, and higher usable blast development rates. Interestingly, no differences in rates of aneuploidy. They did not look at mosaicism. That'd be an interesting endpoint to look at. And Mohammed Fazi out of Egypt has also done a very good temperature study looking at not quite a large temperature difference. I think he looked at 36.5 degrees versus 37 degrees. And again, showed an absolute impact on embryo development. So certainly an area of active research as we continue to explore embryo physiology and see what we can do in the laboratory to improve outcomes. But it is important to note that embryo culture media and most of what we've done has been tested and trialed at 37 degrees. And so perhaps not surprisingly, we see better outcomes here because this is how media was formulated and how we have done most of our experiments in trying to optimize our environment. That being said, temperature is very hard to maintain in the lab. And so this is from an older publication in JARG, kind of a landmark paper from back in 2002, where they used some real-time temperature measuring devices, and they looked at various surfaces and warmed environments in the laboratory. And the take-home point here is none of these lines that you'll see are straight lines. You can see they're all drifting down. So cooling occurs very quickly and very readily in the laboratory. So things like media volume, use of oil, use of lids, type of incubator, type of dish, presence of stage warmers are all things that we use in the laboratory to help maintain this temperature. And to do that, you want to make sure that you measure temperature to know what you're getting, and then take precautions to help maintain that temperature. So first and foremost, set an acceptable range in your laboratory. That's for both surfaces and incubators. Use calibrated devices. If you don't have a calibrated device, it's really not useful if it's not accurate. Measure temperature in the media There can be some differences in temperature in the media versus the surface based on the type of dish or other factors. You want to measure temperature regularly, at least daily. You can use data loggers, and then take steps to maintain or stabilize the temperature. Lids, oil, aluminum blocks, a variety of other methods. And really then, the other things that we do in the laboratory in terms of embryo manipulations, we want to be aware of and make sure that we're not overly stressing our gametes and embryos through mechanical stress. And so there are obviously some studies that show less embryo handling and use of uninterrupted culture can benefit embryo development. This would suggest that less mechanical stress or the associated stresses like we talked about, temperature and pH, can improve embryo development. We know that embryos can sense shear stress, and so repeated moving between media or repeated pipetting can be damaging. And so really, by and large, fewer manipulations outside the incubator can help avoid pH and temperature deviations, can avoid shear stress, can avoid potentially losing embryos, less handling, less risk of things going wrong. And now there are papers coming out, as you can see here, trophectoderm biopsy protocols can affect clinical outcomes. So how we're biopsying these cells, and certainly can be a traumatic experience, and some techniques are better than others. Doing biopsy at different stages, as we mentioned, cleavage stage embryos are sensitive and blastocysts are a little bit more robust. And so how we biopsy, when we biopsy, and even with ICSI, there's now publications and certainly historical publications talking about various ICSI techniques. And I think this is an area that we're going to hear about later, I think, from other speakers. But there are groups looking at use of artificial intelligence and or automation to help not only optimize these protocols and really take out that variation and try to improve upon what we're doing manually in the laboratory. So I think this is an area that will, in the future will greatly help us. And so in conclusion, there are various variables in the IVF laboratory that can impact embryo viability and outcomes. We've talked about things like pH, osmolality, temperature, and others. And really, we want to make sure that we minimize the amount of stress that we're subjecting our gametes and embryos to. And that means taking appropriate steps to maintain that environmental stability, reduced handling when possible. And importantly, there's still a lot of work to be done. So a lot of research left for us to do to continue to improve upon what we do. Thank you very much.